Good evening, 6th graders. This is Mr. Coranza. Let's talk about lesson 12 on genetic variation. Genetic variation, as you're going to read in the lesson, is the direct result of sexual reproduction. You cannot have genetic variation without sexual reproduction. That is a fact. Now, because it's sexual reproduction, we know it involves two parents. And when we have two parents in the mix, we're going to end up with different combinations of different versions of the same characteristic. In this case, we're looking at these frogs. They have different patterns, different colors in their skin, and they very well could be related to each other. They could be siblings or cousins, but because they belong or they come from parents, two parents, they're going to have a combination of the parents' alleles, of their parents' genes. And we can see very strikingly, they have different phenotypes, but we can infer, we can assume that their genotype is also different. That's where the word genetic comes from. They have different genes, different alleles, different versions of the same genes, and the word variation comes because they vary from each other. They're different from each other, and that's when we have animals or plants or any type of organisms that are related to each other, but they vary genetically. They are different from each other, not only in appearance, but also in their genetic makeup, genetic profile. Now, if we keep going, we're going to see some examples of genetic variation. So in this case, we have a group of animals. They're moths. They're called pepper moths. And they are related to each other just in the same way that the frogs were related to each other, in the same way that you and me and other students are related to each other. So they belong to the same species, and they look different. So very instinctively, we're gonna say that they probably reproduce sexually because they look different from each other. They're not exact copies of each other like we would expect in asexual reproduction. Now, there are some advantages of genetic variation, and the advantage that is gonna be covered in this lesson is the advantage of survival. So what better way to see it than look at it directly? So in this case, I'm gonna pose this little game, and we can see that even though the moths are related to each other, as we saw in the previous image, some of them are black. We cannot really tell them apart from the tree bark, and some of them are white. Now, if we have predators in the environment, in this case, let's say pigeons, the pigeons are gonna eat the white moths really fast, and they're gonna have a hard time picking up the black moths. So the bird is not going to be able to really eat them. And because of that, because of the difference that they had between each other, some of them might die, but some of them are going to survive. And that's the key advantage of genetic variation, is that we're going to have differences in organism, but because we have differences, we can also have better chances of the species surviving, of individuals surviving and having even more kids. In this case, a lot of them survive, but there were the black moths in here. Another white moth, I'm gonna eat it. So we're gonna have the same example in here. It's just individuals surviving because they were different from each other. It increases the chances, and that's something we're gonna observe in every animal or every plant that you can think that reproduces sexually. They're gonna have little difference between each other, and they're gonna have an advantage compared to asexually reproducing organisms. That if the environment changes, is this climate where the chickens live becomes hotter, or a predator comes, and some of them might camouflage with their environment, or some of them might be able to resist the higher temperatures or the colder temperatures. Some of them might die, but some of them might also survive. So in reality, even though some of them might die, the whole species, the group, is gonna survive because some of them were better prepared for the changes in their environment. So that's something we're gonna keep pushing next week, but we just wanted to be introduced to that concept, okay? And the key wording here is survive, reproduce, and pass on their genes, okay? So keep in mind these three concepts. Genetic variation, because they're different from each other, is going to allow some individuals to survive, reproduce, and pass on their genes. The second example, very briefly, and it's a very sad example actually, is the Tasmanian devil. So we might know them better from the Looney Tunes cartoons, but Tasmanian devils are a species that they come from Australia, an island close to Australia, which is called Tasmania, and they are not very different from each other. They are because they were hunted down, they were persecuted, and their habitat has been destroyed so much, they are all very similar to each other, not only in appearance, but they're also genetically almost the same. They're like as close as we can get to copies from each other's. Now, the disadvantage of this is that if the environment changes, or let's say a predator comes, because all of them are the same, they're probably all gonna die. The species is gonna die, the group is gonna die, and that's exactly what happened when a disease appeared. The disease, as you can read in the reading, is a contagious tumor. It's a contagious cancer. We don't really have contagious cancers in humans because we're very different from each other. But these Tasmanian devils, because they're so similar to each other genetically, their genotype and also their phenotype, the disease just spread out like wildfire between them. They ended up all having this tumor, and the species is close to dying in maybe 5 or 10 years because they die. It gets spread out just like we see some diseases in real life, and the whole species might die because they didn't have the advantage that these moths or these frogs or even us have, which is the genetic variation. Right, so we can see how important and how crucial is genetic variation for the survival and the survival of the species when the environment or diseases come or the environment changes. Again, we're gonna keep pushing through that idea, and it's something we can see in different species. Now let's go through the activities. So for this week, you have 2 activities, 2 practices rather. The first one in here, you can see it's a Google Doc. You're gonna click on it. You will find it over here in your lesson 12, and it's a Google Docs. It's about generic variation. Please read the just read the first abstract, whatever comes before the activity. There are some questions. You're gonna, just the same as you did in the survey, you're gonna place an X where your trait, your allele comes, right? If you have earlobes that are loose instead of attached, you're gonna put an X. Your hair type is curly instead of straight, you're gonna put an X. So whatever you end up with, you're gonna write it down. You have in total 7 traits, and what you're gonna do is in this table that you see in here, which is a little intimidating, you're gonna end up finding your number, and by that, let's look at an example. So depending on what trait or what allele you end up, you're gonna write down the letter that comes after. Okay, the alleles. In this case, dominant alleles are together with heterozygous, so dominant alleles only have 1 letter, which is fine for the activity that we're doing, and you're gonna write them down. After that, we're gonna find our number. So if you read the example before, it just gives an example. This person is gonna have loose earlobes, curly hair, curling tongue, no hair on fingers, dark eyes, no peak, widow's peak, and a straight thumb instead of a hitchhiker thumb. And they say the genetic number is 108. Let's find out how they found 108. So let's start by loose earlobe. So the first letter, we're gonna start in the middle of the disc, in the middle of the circle. We're gonna choose either loose earlobe or attached earlobes. Let's choose loose because that was the example. After that, it gives us 2 options, either straight hair or curly hair. We're gonna choose curly hair. Then it's gonna give us another option, curling tongue, or you're not able to curl your tongue. We're gonna choose curl your tongue. After that, we're gonna put no hair, fingers. We're gonna choose capital A-E because dark eyes. By the way, we're still using the example right above it if you following with me. Then we're gonna end up with no widow's peak, so we're getting close to the outside of the circle. And then we're gonna go with straight thumbs instead of hitchhiker thumb. And that's when we're gonna end up with the number of 108. Okay, so we started with here, and we made our way outside the circle just by choosing the traits that we had. Right, and in this case we went all the way to 108 just by choosing the 7 alleles, the 7 variations of our trait. So that's a little fun activity that you might see, and you can see just by choosing 7 characteristics, you can have 128 combinations. So that's a really big Punnett squares that we could make. We're not gonna make a Punnett squares this size, but you can see just by choosing 7 characteristics instead of the usual 1 that we usually do, you can end up with 128 combinations. And we're not really counting heterozygotes in this case. We're really doing either dominant or recessive. So 128 is a huge number. Now think about this. We're only looking at 7 genes or characteristics in here. We have 20,000 characteristics or genes. Now think about the number of combinations we can do with 20,000 genes. It's pretty big. We wouldn't be able to count it, and scientists agree that the number of characteristics is that you cannot end up with 2 people that are exactly the same. It's impossible. Alright, so you're gonna put the number in here when you find it, and after that you're gonna answer this question, and then you're gonna do this reading. It's about the deer. Some of the checking questions from this week are regarding this reading right here, which is all about the white-tailed deer. It's a cool reading. It's about a species that lives in North America. You're gonna answer this question. You're gonna think if they live in North America, close to New York, is that temperate climate or tropical climate? Hint, hint. And then we're gonna do this game. So a lot of people have been asking for the clues for this game, so let's go through it. Thank you for the people that have been reaching out. You are very cool, very awesome. So let's look at the instructions. So we have some cards in here. These are our deer. So this is our pack. This is our group. We are part of this group. These are our brothers and sisters, and we're gonna have a total of 10 of them. Okay, so they are a pack. We're gonna see if we can survive the winter. So the instruction says, first of all, roll your dice, and then after you roll your dice, we're gonna pick the letter according to our die roll. So what better way to exemplify that just by doing it? So if you don't have a die, don't worry. If you can click in here, it's gonna take you to Google Dice, and we're gonna roll our dice. The number is 3. So we're gonna look at number 3, and it says the young fawns. If you do the reading, you know fawns, and I'm gonna give you the answer. Fawns are the baby deer, are particularly vulnerable to predation by coyotes. A fawn with a poor sense of smell might not be able to detect a coyote in time to escape. So we can see right there, they're talking about a characteristic, rather an allele, a variation of that characteristic. Some fawns, some deers do not have a very good sense of smell. Some of them do have a good sense of smell. And it says, lose one white-tailed deer that has poor sense of smell. So we're gonna go to our cards, and we're gonna look at their characteristics. So we're gonna see poor sense of smell. A couple of them have good sense of smell. That's the third characteristic in their list. And some of them have a poor sense of smell. We're gonna choose one just like the letter card says, and we're gonna place an X because they are not gonna make it. Sorry, deer, but you're not gonna make it this turn. Now we still have 9 deers. Don't worry. So what we're gonna do is we're gonna repeat these 3 more times. So 3 more times. You're gonna choose 3 more cards after that, depending on the number. Let's just do one. I'm not gonna go through that one, but let's say we roll the die again. It's gonna be number 6, so we're gonna go to number 6, and it says lose one. Please do the reading. It's very interesting, and it tells you why they're not gonna make it. But it says at the end, lose one white-tailed deer with a small home range. So once again, you're gonna go here, and you're gonna look at the 4th trait. Most of them have a large home range, but some of them have a small home range, just like this one. We're gonna place an X, so this might not make it. And we already did it 1 more, so you're gonna do it 2 more times. After that, hopefully you end up with some deers that are left. Hopefully some of them make it, and I have a good feeling that they're gonna make it because they're very distinct from each other. Once again, they have genetic variation, and that means that they have an advantage that other animals or other species or organisms that don't reproduce sexually are not gonna have, which is that even if the environment changes, even if we have some predators like coyotes or hunters, or it gets really cold or it gets really hot, they're different from each other. Some of them are going to make it, and eventually those that make it are gonna be able to survive, reproduce, and pass down their genes. Remember that word from here? Survive, reproduce, and pass down their genes. So I'm not too worried about those deers. I think they're gonna make it, and after they make it, you're gonna answer those questions that are in here. It's gonna ask you some questions. How many are left? Do you think what characteristics they had that made them, better or more prepared to survive? And you're gonna answer those questions. It's a fun game in my opinion. I did it a couple times. If you want, after you do the questions, try to up the number. So try to roll your dice more than 3 times, more than 4 times. Let's see how many turns, how many changes it takes for them to just completely being wiped out, just like the Tasmanian tigers. But that's if that's your option, if you want to do it after you do these questions. All right, now let's go to the last activity, which is the practice. This practice is more about you looking at what the result of putting data in tables looks like. So if you go to a lesson, we have different tools that scientists use to look at data, and you're gonna do this definitely in math later on. In statistics is something that we call, you're gonna see it yet, but it's something that you do in math and science a lot, which is just making graphs. And graphs are just a visual way of you looking at your data. So whenever you're gonna do an experiment, you usually collect the data and numbers, but sometimes this number, they don't tell you that much just by looking at them. So we put them in graphs, and just in this graph alone, we can see that whatever this middle group is, it's very high, very tall. So we know that this middle group is gonna have more students than the other two. We're gonna know just by looking at this. We don't know the numbers, but we're gonna know that this group is very small. And if we go back to the table, we know it's exactly what we see. The middle group is gonna have 56 students, so that's why we see that it's so tall. And we're gonna be able to very observe our just visualize our data in that. You can do different colors, you can do different types of graph. And speaking of different types of graphs, the other one is called pie chart. It's another cool chart that is gonna be is gonna show you groups. Instead of looking by height, it's gonna tell you the percentages. Just by looking at this, you know, when it has half of the circle, it's 50% of the students. Another one is 30% of the students, another one is 20% of the students. So it's just another cool way to view your data. And when we go to the practice, what you're gonna do very simply is just input the data that is in here. So a number of students you're gonna put in here. So the first group is gonna have 2 students, then you're gonna put 16 students, then you're gonna put 16 students again, you're gonna put 23 students, and you're gonna put 2 students in here. And you're gonna see how the data shows in the table. It's really cool to look at it. In the last example, you're gonna put your own trait. You can do it with your friends or family, but you're gonna choose a characteristic. Let's say we look at favorite food. I messed it up, but you can put favorite food. You can put whatever you feel that you wanna show, and you're gonna put the number of people in here. And when you put it, you're gonna be able to see not only the pie chart but also the bar graph that you're gonna end up, which is something that we're gonna keep practicing. And it's really cool to just look at data visually instead of seeing it in numbers, because numbers can be a little intimidating. We cannot really make a lot of sense out of it, but when we look at it in this way, we know this group, whatever it is, this green group is definitely bigger than the others. One last thing that I want to point out is that if you go to the results of our survey, you're gonna see that we ended up having a lot of 70-20%. Just like we were doing in the Punnett squares, we ended up having the same variation that we did in the Punnett squares in real life, in our classrooms. If we look at this one right here, this is actually almost exactly 75%, 25%. So whatever we were doing in the Punnett squares is clearly something that we can observe in humans. If we maybe did this experiment or this survey in the entire school, or entire New York, or every person on the planet, we might end up with the same percentages. So it's something really cool to think at, is that whatever we're doing, Punnett square, heterozygous crossings, homozygous crossings, is something that is based on real life. And the percentages that we get are really what most likely, or the higher chances of us getting in real life. So that's what I wanted to end up with. If you have any questions about the practice, about any type of the reading, any type of questions about genetic variation or the assessments or anything that you want, please let us know. I'm gonna end up posting a review sheet later this week. Please look at it. If you finish the practice early, please go through the review sheet and try to see what things you might have forgotten from the past lessons. We're gonna eventually test you on them, like a quiz or a test, so you know that you have retained, you got that knowledge down, and you're able to just pass any type of test that has anything to do with heredity or genetics. or genetic variation, or any topic that we cover. That's what review is for. That's what this week is for you to catch up on your work, and please ask questions if you have some. Thank you, and you have a good night. Bye bye.